Hello, everyone. This is Kun Zhang from Xavier University of Louisiana. It's my pleasure to present our recent research about polygenic basis of racial disparity in prostate cancer. If you are interested in this work, you can refer to the published article in Cancer Prevention Research. As we know, multiple racial disparities in prostate cancer have been reported to date. For example, prostate cancer is more prevalent and aggressive in African Americans in terms of higher incidence and mortality rates. However, among the 150 reported risk SNPs for prostate cancer, less than 10 can be identified from available data collected from African ancestry. Therefore, relevance of those reported risk SNPs to prostate cancer disparities remain to be determined. So in this study, we hypothesized that polygenic risk score is an ideal marker to assess such a relevance, and a high-risk population such as African Americans has a higher average PRS. To test this hypothesis, we retrieved 3 GWAS data from dbGaP, prostate cancer data from TCGA, and sleep data of African and European subjects from 1,000 genome. We used those GWAS data for model training and the other datasets for testing. In our study, we employed those 3 methods to compute PRS based on 93 reported index SNPs for prostate cancer. Here are the PRS prediction on 1,000 genome samples from African and European superpopulation and their affiliated populations. In plots A, B, and C, PRS are calculated according to the test anneal effects that are estimated using each of those 3 datasets. Plot D presents the aggregated or average PRS obtained over 3 datasets. Those 2 plots show the stratification of aggregated PRS on TCGA prostate cancer patients. Plot A is across racial groups. It's clear that Black patients have higher PRS than white patients. In plot B here is over somatic mutation-based cancer subtypes defined by the TCGA research team. In plot B, the 4 category on the left constitute the ETS fusion positive subtypes. The rest form the ETS fusion negative subtypes. It's evident that the patients with fusion in ERG and other ETS family gene in general have lower PRS than the patients without such aberration. This difference is statistically significant. Using TCGA samples, we further scan the correlation between patients' aggregated PRS and gene expression. With p-value cutoff at 0.01, we identified those 10 significant genes. In this study, we also evaluate the consistency of predicted PRS using another 2 methods. So, in summary, our study shows that polygenic risk score is an ideal quantitative marker to relate reported risk SNPs to prostate cancer disparities. And a high-risk population, such as African Americans, has a higher average PRS than no-risk population. Our study also shows ETS abnormal cancers may be less heritable compared to other subtypes. So for detailed information about our research, please refer to this article. Thank you so much.